everyone, this is Dr. Yoder Himes. We're going to be talking today about microbial symbioses, and I'm going to be talking about one particular microbial symbioses. The point of this video is to provide a supplementary instruction for those of you who are not attending the active learning TIL session regarding this topic. And for the homework associated with this topic, you'll need to go into the literature and the internet and look up various symbioses that occur between microbes, including bacteria, archaea, and fungi, and their hosts that you have listed on your homework, whether it's plants, marine vertebrates, insects. I'm going to be talking about one specific one that I've done some research on, and I'm going to show you kind of what I'm looking for in terms of some of the things we should be thinking about when it comes to microbial symbioses. So let's talk about this particular symbiosis. This is a symbiosis that occurs between a bacterium known as Xenorhabdis, Nematophila, or Nematophilus, which is a gram-negative rod, facultatively anaerobic, and a worm. In fact, a little worm called a nematode. And these are just little worms that exist in many different natural environments, waters, soils. And this particular nematode that forms an association with Xenorhabdis is known as Steinermina carpocapsae. So this is a particular nematode, and it's a special nematode because it's known as an entomopathogenic. Okay, and as the name suggests, this particular nematode can actually cause a pathogenic interaction with insects. And the way it does that is what I'll describe next. But one of the features of these nematodes is they actually have an organ that lies fairly close to their mouth. This is called the bacterial receptacle. Sometimes I've seen it called a vesicle instead. And it is just densely packed with this particular bacteria. So this is kind of like its tote bag that it is carrying of these bacteria. Now this nematode, as I said, it's a pathogen to insects. And it's a pathogen to a variety of different insects, including caterpillars, crickets, grasshoppers, worms, butterfly. We're going to draw its life cycle here. These are called infected juveniles when they are carrying Xenorhabdis nematophilus. Infected juvenile. And this infected juvenile stage, all it does, it doesn't eat, it doesn't sleep, it doesn't do anything except search for a new host. We're going to draw its host as one of these fat little waxworms. And waxworms are pretty simple organisms. They have a mouth. They have an anus. And the nematode can penetrate either of these. Okay? The infected juvenile. And it needs to do this as part of its life cycle because it cannot reproduce outside of the insect host. After penetration, the infected juvenile secretes some of the bacteria into the hemocoel, which is kind of like the internal goo inside the waxworm or the other insects. And it's going to spill this out. And these bacteria are going to start rapidly growing. Okay? And as they do so, Xenorhabdis produces a variety of compounds which will actually end up killing the insect host. So they produce hemolysins, cytotoxins, which will all help in a large variety of proteases, lipases, which break down lipids. They also produce pigments, which I'm not sure what has to do with anything, but you can actually tell a infected waxworm versus not. And all of these degradative enzymes then are going to wind up turning this organs and things inside the insect into a big, gross, mushy mess. And that is the mushy mess that the infected juveniles can use to start their growth and reproduction cycle. So one gets in and replicates within the hemocoel of the insect. Eventually, this will lyse the waxworm or the insect, releasing infected juveniles and lots of bacteria into the environment, where they can then proceed to look for a new host. Okay. The other thing that Xenarabidus does is it produces antibiotics. It produces antibiotics to keep away other bacteria during this process. As you can imagine, as this waxworm dies, it becomes susceptible to degradation by lots of microbes. But the Xenarabidus, oh, I did it up here too, Rabidus, produces antibiotics to keep this environment clean, and so that it can dominate in the hematocele or the hemocoel, and then can be the dominant organism that colonizes the bacterial receptacle in the newly replicated infected juveniles. Okay, so this is kind of the life cycle. This, in general, this process here takes about 1 to 48 hours to occur. So from infection time to lysis, 48 hours for this. So let's think about this in terms of the microbes here. So the benefits. This will allow us to figure out what type of symbiosis this is. So obviously to the microbe, Xenarabidus, they're getting nutrients during the replication stage here, when they start lysing and degrading all the insect proteins and nutrients. The microbe also doesn't have to worry about living outside of a host. It can live happily in these infected nematodes, and this can allow for dispersal into subsequent animals. It also, because of these things, especially the nutrients, the bacteria can reproduce. Okay? So those are some benefits. Now, note here that Xenarabidus can actually live outside of the host. So it is considered a facultative symbiont. Okay, so let's talk about what happens to the host. The host in this case is Steinoderma carpocapsae. So this microbe allows this nematode to reproduce. And in fact, without Xenarabidus, this particular nematode cannot reproduce because it cannot sufficiently break down the nutrients inside the insect host. So to it really has to have these microbes in order to complete its life cycle. Now, it won't die if it doesn't have them in the infected juvenile stage, but remember, these do not actively grow, reproduce, or eat. So this would be a terminal step if it did not have the Xenarabidus inside of it. Okay, so because of this, you can see that these are all beneficial and these are all beneficial, which suggests that this is a mutualistic relationship. Mutualistic symbiosis. Okay, so now we need to think about the abiotic factors and biotic factors that drive this evolution of this particular symbiosis. So, certainly the abiotic factors that influence us are nutrients. nutrients for both the host and the microbe in terms of whether they can find available insects which allow them to both complete their life cycles. I could also imagine a world in which temperature plays a major role in this types of symbiosis for multiple reasons. First of all, there's going to be a limited range of temperatures that the nematode can tolerate. And while nematodes are generally quite good at surviving in a variety of conditions, too hot of temperatures would certainly influence the evolution of this pair, because perhaps during stressful times, this nematode might instead use the bacteria in its receptacle for a food source. Now, it turns out that actually doesn't happen, because these bacteria can also kill the nematode if I think they get too stressed out, and produce certain proteins to do that. I could also envision a world in which temperature may influence the availability of prey or insects that this nematode can infect. So if you have global climate change, which is driving temperatures up, perhaps that is not going to be so good for the survival of many of these insects. And then they in turn, since they are required for the life cycle of these organisms, that this could put evolutionary constraints on reproduction of these organisms. Certainly these things normally live in soils, so you're going to have constraints that you need to have water and oxygen, because nematodes are animals, or at least eukaryotes. They need oxygen for the most part. So I could consider those as major abiotic factors that are influencing the symbiosis. There's a lot of biotic factors, I think, that are also driving this. So insects have a surprisingly well-developed immune system and can, for some time, influence which bacteria get reproduced in here, which are the ones that get infected. So if you have mutations in this particular bacterium that it becomes now susceptible to the host immune response, those will get wiped out from the population. So this directly influences the evolution of these microbes, because they in turn dictate the life cycle of the nematodes as well. Additionally, the Steinonurma must have an immune response to hold the bacteria in check in the bacterial receptacle. Okay, so I think that that would also be selective for which microbes do well. And interestingly, there have been some genome studies that have been done looking at Xenorhabdus and the closely related Photorhabdus, which infects a different type of nematodes. And it turns out that within a bacterial receptacle, usually you have a single strain of Xenorhabdus, and those differentiate quite significantly with along a species gradient, suggesting that these two are co-evolving together over time. Additional biotic constraints might include microbial competition or nematode competition. If there's other types of nematode that are also looking to infect these hosts, that could play a significant role, or predation in an environment. So if you have so the things that are predators of nematodes include mites, insects, and even things like everybody's favorite, the tardigrades, the water bears. So if you have all of a sudden a bacteria that can no longer produce chemicals that make these nematodes taste nasty to the predators, those are going to be animals that are more likely to be digested. And so there's a selective pressure for these microbes to also convey a selective advantage to these nematodes by producing things that make them taste bad to these types of biotic factors or these predators. So this is how I would approach any particular assignment when it comes to microbial symbioses, and I hope you can use this to work on your particular symbioses for this homework.